Okay, now actor is gonna do mount a completely assembled micro flow cell onto the microscope body. Typically, those flow cells will contain a reservoir where we store all of this reactant solution, and then usually they will place this reservoir here on this little stage. The other side of the entire cell connects to a syringe, and that syringe goes to a syringe pump. This is how he clamps down the syringe to the syringe pump. You will have to first put the syringe in one of the dented area. You could choose any one between these two. They are identical, okay? And then you will have to use this one to fix this entire syringe body into this dented area. The bottom of the syringe need to go to here, and then clamp down to this vent. This way, this part of the body and then also this part, they are both anchored. The flow cell is mounted on one of these ring holders here. The idea is to place the channel right on top of the objective lens. Once this flow cell is in position, we'll use these two metal clips to clip down the flow cell against this ring stem, so nothing is moving. Okay, everything is fixed. And then also, typically when we prepare the flow chamber, we will use epoxy to make the seal. Try to avoid the contact of objective lens to the epoxy layer, because the objective lens, it is very delicate. Epoxy can potentially damage the front element of the objective. Okay, so moving to the next step would be turning on the CellSense computer on the microscope. And of course, the CellSense is one of the console that controls both the microscope body and then also the camera used for fluorescence detection. In order to do that, what we need to do is to first turn on the power supply to the camera. Okay, the power supply is located here at the back of the camera. Once you press this button, you are hearing the fan noise and then also a blinking light right next to this power button. This is the heating process, indicate the heating process here. Okay. We will wait until the flashing light turns itself off. That means the self-checking process is completed. We have to do this, or otherwise the CellSense program on the computer will not recognize the camera. So we'll have to wait until the blinking light stops, and then we can proceed forward. CellSense program looks like this. This is the CellSense program. Double clicking it will open the software. Another thing that is worth noting is the camera selector that is located over here. We have this selector labeled. One is called left, another called right. This directs the light into the different detection paths. Currently, the camera we use for single molecule experiment, it is located on the left side. So if we switch this selector to the left side, then light will actually get into that ultra-sensitive camera. The right side port right now, it has no camera. So switching this selector to the right side of the channel is effectively shutting down the light that gets into the camera. So before you finish all of the preparation work, you would want to actually put this selector on this right side port so those light will not get into the ultra-sensitive camera and then potentially saturate this entire detection. Because this is too sensitive, this over light in this room could easily saturate the whole thing and then also potentially damage the sensor there. So while it is not in use, it is always a good practice to move the selector to the right side. Okay, so this is how this interface looks like. This is a self-sense. When you first turn it on, it will probably pop up some of the warning signs asking you if you want to do some of the auto cleaning. You can just simply ignore those warning by clicking the no. In this panel, you can click the live button and a window will automatically pop up. This is the live view of the camera. And of course, right now, there's nothing shown here. It's completely dark because we put the selector on the right side port. It is directed, all of the light up directly to the other side of the microscope body. So in this window, we're not seeing anything. And then as soon as you actually put this selector from the right to the left, you will start to see light. And then before doing that, we will have to turn off the light in this room so everything will turn black. Okay, so before, and of course that's going to just interrupt our video recording. So we will cover a bunch of other things before we get to that point. There are a few of other things we need to pay attention to on this console. So over here, the most important thing is to check the icon that is on the top right corner of this entire screen. This is where it controls the internal mirror selection of the microscope body. If you select it, click it, you will hear some of the sound, a motor sound inside of the microscope body. That means the internal mirror, it is rotating. And when you see an icon that shows an eye, that means all the light that got redirected into the eyepiece. You'll be able to view the image through the eyepiece. Under this mode, no light will actually get into either of these two ports. Okay, none of these two ports will sense any of the light. All of the light will be redirected into the eyepiece. Pressing it twice, you see both the eye and then also the camera. So this is a 50-50 splitting of the light. 50% of the light, meaning that 50% of the light goes into the eyepiece, and the other 50% of the light will go into this camera. Okay, and then of course, if you press it for a third time, it will go back to the camera mode, meaning that all of the light will get into the camera. For our single molecule experiment, because the nature of being single molecule, the amount of the photons generated from the catalyst will be very few. So by default, we should select this camera mode and then let 100% of the light, all of the photons basically, go into the camera. Okay? Unless we're doing some other type of detections, we will not select the eyepiece or the 50-50 splitting. Okay? But be sure to check this icon, because if it's not in the camera mode, it is accidentally set to the this eyepiece mode, you will not see any of the signals on your computer. Okay? We also have a shutter button over here. Okay? So this shutter button also controls an internal shutter inside of the microscope body, and then we always need to put it in the open position in order to see the light. Okay, those are the things we need to talk about. Regarding the pump setup looks like this. You need to click the main, then later on click the setup button. Over here it will have a few of the options. Now after is doing this operation, he is selecting the volume of the syringe here. He is selecting the total volume of the syringe. Since he is using a 5 ml of the syringe, he selected 5 ml. Click save, we'll save the setting. Operation window tells you how fast you want the string to go. We use the constant flow mode. It will allow you to input the amount of solution you want to withdraw over a 90-minute period. So from there, the flow rate is determined. Save the settings, and then click run. Then this syringe pump is in action. What we are doing is we want the syringe to suck the solution from the reservoir. We do not inject, but we actually suck the solution from the reservoir. Okay, so that's pretty much all before we actually start to see the sample. Another thing we need to talk about before we turn off the light is the focus adjustment. The focus adjustment controller is here. This is only place we can physically adjust the focus of our microscope. Okay. We have two sets of the knobs. One is on the left side, and one is on the right side. They are identical. They do the same thing. The bigger one is a coarse adjustment, focus adjustment, and then the smaller one is the fine focus adjustment. If you rotate the knobs like this, if you look at from here, it is counterclockwise, adjusting the counterclockwise, then the objective will move up. Actually, it is indicated the direction